Prokaryotic cells can be sorted out by shape, and they come in three common cell shapes. The first one is the spherical one, spherical, and it's called a coccus. The second one is a rod-shaped prokaryote and called the bacillus. And the third one is a short and rigid prokaryote that has a spiral shape. It's a spirillum. Many cocci, that's the plural form, forms the streptococci or streptococcus, and some of them form grape shape, and they are called the staphylococci or the staphylococcus. All prokaryotes share some of the characteristics that they are common for all of them. Let's draw a prokaryotic cell, and the green represents the cell membrane, and inside we have the DNA material, the chromosome or the hereditary material, genetic material. It's a circular hereditary material. Besides that, it might contain plasmid, not necessarily all of them, but some of them can contain plasmid that replicates independently of the chromosome. The yellow one is the cytoplasm, that everything is embedded in the cytoplasm inside of the cell, and these dots are going to be the ribosomes that they are responsible for the protein synthesis. Their ribosomes is a little bit, the prokaryotes ribosome is a little bit different than the eukaryotes ribosome. We will get into it later. The outer purple stuff is a cell wall. Nearly all of the prokaryotes have cell wall, and the cell wall provides physical protection for the organism, and it can be stained by Gram stain, and it can be positive or negative. The positive, that means it picks up the stain and doesn't let it wash out, so we call it a Gram positive, and it means thick wall. The negative has a thin cell wall, and it's worse in pathology, and it's more toxic than the positive bacteria. Now, the cell wall is made up of peptidoglycan. Peptido, it's made out of peptide, and glycan refers to the disaccharide part. So it's a complex macromolecule. The orange circle represents the capsule. It enables prokaryotes to adhere to their surface, to their substrate, or to other individuals in a colony, and also shields some pathogenic prokaryotes from attack by the host cell. The flagellae are for motion. Not all of them contains, but they can have, and as well as they can contain fimbriae. And this is for sticking to surface or to each other, or latch onto the host cell as they colonize the host cell. The population of the prokaryotes, they adapt rapidly to change in the environment, to changes in the environment. Prokaryote population happens. They divide by binary fission, and within an hour they replicate, and 1 will be 2 and 2 will be 4. Because of the rapid population growth, there's the spontaneous mutation creates a high level of genetic variance, and that increases the survivalship of the individual bacteria when the environment might just go through a change. The great deal is that many prokaryotes form specialized cells called the endospores. They form endospores, and that remains dormant through harsh conditions. For example, hot weather, cold weather, or lack of water. Endospores can survive extreme weathers. When the endospore receives environmental cues that conditions has improved, they can absorb water and they resume their growth. Going back to the prokaryotes, now we call the prokaryotes that they live in extreme salty environments, halophiles, extreme temperatures, or in acidic pools, they are thermophiles. And methanogens, do I have the methanogens? Yes, I do. They live in anaerobic conditions where there's no oxygen, and they give off methane as a waste product by digesting the cellulose in the cattle's and the deer's intestines or stomach, in the digestive tract, and as well as they decompose materials in landfills. They produce methane that is a very strong greenhouse gas, and because of the greenhouse gas, the global climate, they facilitate the global climate change. And that is sad.